Hello, my name is Charlotte Hemby,
And I'm Liz Parker.
and our research is on the presence of antibiotic resistance in soil bacteria. So our abstract, we wanted to focus on one of the most prominent health concerns, which is infectious disease caused by antibiotic resistance. So outside of medical settings, bacteria in different environments are starting to show high levels of antibiotic resistance and antimicrobial resistance. So our research was focusing on what bacteria is resistant to 8 different antibiotics and if they produce any antimicrobial properties.
So I'm going to do our introduction. And we found that it's been very common over the years for um, antibiotics to be overused and oversaturated in many different areas. So in the healthcare system, they are prescribed very, very frequently. And it has led to these antibiotics and antimicrobials going into the soil samples or going into soil, period. So we found that through the CDC, antibiotic resistance is when bacteria and fungi develop the ability to defeat the drugs that are designed to kill them. So it basically means that these defenses against bacteria and illness-causing pathogens have been, or pathogens, are not effective anymore.
So for our methods, we gathered 4 different soil samples across Raleigh, focusing on different areas adjacent to bodies of waters and the exposure to sunlight. So our locations were Lake Raleigh. So we did one that was on the embankment of water, and then one that was a few feet away. And then we got 2 other soil samples at Meredith College, one that had full sun exposure and one that had no sun exposure. So after collecting our soil samples, we then um, did a serial dilution to cultivate small bacterial populations to work better, to see what we're working with. And then after we completed that step, we T-streaked to isolate different bacterial colonies, so it's easier to work with. And then with that, we followed it with gram staining, to see if the bacteria was gram positive or gram negative, which is very crucial because gram negative bacteria have been shown to be more resilient to antibiotics. So once that gram classification was established, then we tested against 8 different antibiotics, and they were penicillin, erythromycin, neomycin, tetracycline, gentamicin, streptomycin, ampicillin, and chloramphenicol. So you can see those in our results, but we also tested for antimicrobial properties to see if the bacteria produce any zones of inhibition. And so we used escape pathogens, and those were Staphylococcus aureus, Pseudomonas fluorescens, E. coli, Klebsiella aerogenes, and Klebsiella pneumoniae. And it is important to note that Actinobacter was not used because of facility limitations.
So in our results, you can see on the first 2 pictures, so those darker rings around the discs are where the bacteria isn't allowed to grow, so the zone of inhibition. Each circle is labeled, or each disc is labeled with one of the different antibiotics. And you can see especially with the penicillin and ampicillin, that the bacteria is still able to grow. There aren't any zones of inhibition, which means that they are resistant to those antibiotics, which in turn means that these antibiotics that we're using are not really effective towards bacteria that's literally in our soils. So the next graph shows we went we measured all of the zones of inhibition and did the standard deviation of those, and just found out a range of how effective these antibiotics were based on the size of the zone. And you can see that in every single category, or yeah, for every single category, there were areas that the antibiotics were not effective at all. And at the last figure, that is our antimicrobial resistance testing. So we have 2 discs, each are 2 different types of antimicrobial discs. And you can see that there are still zones of growth around those discs, regardless of the fact that they're there in the first place. So that just shows that they are still able to grow and resistant to antimicrobials.
So for our discussion, out of our 4 samples that were collected in Raleigh, the near water and away water samples showed that for the class of beta lactams, which are penicillin and ampicillin, shown in figure 1, show no zones of inhibition, meaning that the bacteria can grow even in the presence of these antibiotics. It also shows no zones of inhibition for streptomycin in our sunny areas. So those are streptomycin is very common. It's a very commonly prescribed antibiotic. And then with our samples, we tested against the escape pathogens. All of our samples showed no zones of inhibition, which is interesting to find because these bacteria actually grow with alongside different pathogens. So there's no competition needed for these bacteria to live in the presence of E. coli, Staphylococcus aureus, and the other escape pathogens.
Yeah, so we found that there is in fact a presence of antibiotic resistance in the soil samples that we found. We also found that it was interesting to see that the soil samples that were collected from the away the body of water showed more resistance to penicillin, ampicillin. All 4 of our samples did not produce any antimicrobial properties at all, as Charlie was just saying. And our next steps are really just getting more students and undergraduate research involved in looking into this and looking into microbiology in general. I think it's that 90% of all undergraduate research or all research in microbiology is by undergraduate students. Yeah.
Yep, so that was our presentation.